Hello, everyone, and welcome back to this new series of INFOGEST Viva webinars. My name is Daniela, and I'm here to present the 14th INFOGEST Viva webinar. These webinars are organized by a team of postdocs from the Food Research Center of Moore Park, Teagasc since 2022. And usually we do them on the first Wednesday of each month with the goal of providing you the most recent findings in the field of digestion from recently completed PhDs. Our speaker today is Jean-Michel Fernandes. Jean-Michel has a bachelor's degree in biochemistry and a master's degree in bioengineering, both completed at the University of Minho in Portugal. He joined the doctoral program in food science and technology at this university in 2020 to start his PhD studies. His research involved work in the fields of nanotechnology, nanoemulsions, in vitro gastrodigestion and absorption, and bioaccessibility as well, to develop tailored functional and excipient foods for the older adult and analyze the digestion in vitro. After his presentation, there will be a Q&A session with him. So I'm inviting you to use the Q&A button at the bottom of your screens if you'd like to join the discussion. The webinar will also be streamed live on YouTube and will be available on the InfoGest YouTube channel afterwards, and you will be able to find a link to the published articles presented here in the video description as well. Jean-Michel, the floor is yours. Jean-Michel, the floor is yours.
Hello everyone, my name is Jean-Michel Fernandes, and today I will present the work that I developed during my PhD thesis entitled The Development and In Vitro Analysis of Tailored Functional and Excipient Foods for Elderly Populations. During this presentation I will do a brief introduction, followed by the thesis motivation. Then I will present chapters 3 to 6 corresponding to the obtained results, and finally the conclusions and perspectives for future work. We know that worldwide population is aging, however, healthy life expectancy is decreasing, and this is mainly due to the loss of physiological functions that mainly affects elderly populations and makes them more susceptible to suffering several types of diseases. As such, it's important to make appropriate dietary choices that can go by the development and consumption of tailored food products to improve their nutrition. We also know that aging is a multidimensional process in which it's difficult to establish an effect and a cause, since everything is intertwined. What we do know is healthier and more nutritious food are demanded, and this can be obtained using different strategies, such as functional foods that can use nano-based delivery systems to encapsulate several types of bioactive compounds. In our case, we focused on lipophilic vitamins for their incorporation in different types of matrices to obtain food products with additional functionality. Other strategy is to produce excipient foods that can use the same nano-based delivery systems, not to encapsulate bioactive compounds, but rather to be co-ingested with nutraceutical-rich foods to improve the absorption of those nutraceuticals already available in food products. However, we cannot only observe the development phase, we must also understand the digestive behavior of these food products in the targeted population if we want to have the food products with desired food properties. As such, there are two main questions regarding this thesis. One is correlated to the fact that there are not a lot of food products developed specifically for the older population. And other is that we do not know for sure how these food products are digested by the population targeted. As such, our research aims were to develop food products tailored for the elderly population, to improve the bioaccessibility of nutrients in deficit, and evaluate the developed products using in vitro gastrointestinal systems. Moving on to the results, we initially studied nanoemulsions and excipient nanoemulsions as α-tocopherol delivery systems, to understand how these strategies would modulate the stability, bioaccessibility, and stimulate the bioavailability of our derived compound. To do that, We developed oil-in-water nanoemulsions encapsulating vitamin E, and the same oil-in-water nanoemulsions without any bioactive compound encapsulated into it to function as the excipient nanoemulsion. The nanoemulsions formulations were optimized using a central composite design, and the formulation selected was then applied to the in vitro gastrointestinal protocol proposed by the INFOGEST. From the formulation optimization, we saw that encapsulation of α-tocopherol was not statistically significant, since both structures had similar outcomes. We also saw that higher concentrations of surfactant and lower concentrations of oil originated smaller particles with better polydispersity, and this was expected since we are reducing the interfacial tension. We also saw that longer processing times originated samples with better homogeneity, and here the high-pressure homogenizer was more efficient than the ultra-Teagasc. Analyzing the particle morphological characteristics during the in vitro digestion, we initially validated that all produced structures had similar sizes, and once more that the high-pressure homogenizer was more effective in homogenizing our sample. Also, during the oral phase, we did not see significant differences, but then when we arrived at the gastric phase of our in vitro digestion, we saw that the excipient nanoemulsions were more susceptible to the destabilization. And this could be due to the fact that they are void of any bioactive compound inside, so they were more prone to destabilize. Regarding the data potential, we validated that the produced nanoemulsions were stable because of the mode obtained, and then throughout the digestion we did not see significant differences. As for the behavior of these structures as α-tocopherol delivery systems, we analyzed the stability as the percentage of α-tocopherol that was untransformed during the digestion. We can see that the control had a higher concentration at the end of the digestion, and this seems like an outlier but can be explained by the fact that there was no surfactant added to the sample. As such, during the digestion there was phase separation that prevented α-tocopherol from being degraded, but also prevented it from being solubilized, as we can see when determining the bioaccessibility, in which any of the strategies applied was statistically significant better than the control sample. Also, by analyzing the simulated bioavailability as the product between these two, you can see that each strategy was better than the free form of α-tocopherol, with particular importance to the nanoemulsions encapsulating this bioactive compound. And from this work, we could conclude that the in vitro digestions with nanoemulsions could have improved the emulsification mediated by biliary salts, hence improved the micellization and α-tocopherol bioaccessibility. In the next chapter, we went to understand the influence of in vitro protocols simulating the adult and other adult gastrointestinal tract on the digestibility of fortified yogurts. To do so, yogurts were fortified with the described nanoemulsions containing α-tocopherol, and adaptations were made to the in vitro dynamic gastrointestinal system DIDGI, in which were proposed conditions for an older adult model, which comprised a prolonged gastric emptying as well as a longer acidification curve, lower concentration of enzymes, and as well as lower concentrations of biliary salts. These protocols were compared regarding the α-tocopherol release kinetics. During the gastric phase, we evaluate the particle size distribution of our sample. We can see that at initial stage, samples presented similar distributions, and digestion was similar until T1/4 of the gastric phase. However, from then, the adult in vitro digestion protocol was much more efficient in degrading our gastric samples, since we can see at orange that the digestion was the digested product was more homogeneous. As an effect, our α-tocopherol release kinetics during the intestinal phase were significantly affected. As we can see, the adult model in blue was much more linear compared to the older adult model, in which it was higher at the initial stages but then decreased. Because of that, the α-tocopherol recovery was overall higher in the adult protocol, with the exception at the point of 3 hours. Even if we don't see statistical significant differences, there was higher concentration in the older adult model that could be explained by the fact that there was still gastric sample being delivered to the intestine. As such, α-tocopherol was available to be released. Interestingly, we can see that once more at initial stages the adult model was more efficient in release of α-tocopherol from the intestinal reactor. However, at the end of the digestion, we see that the proportion between α-tocopherol delivered from the intestine and inside the reactor was similar in both protocols, which can suggest that even though if we prolong the gastric phase and reduce enzymatic activity, both protocols were equivalent in degrading the product. And this can be also observed in the bioaccessibility, which there are no significant differences rather than the point at 3 hours by the motives explained before. From this, we can conclude that the non-emulsions stabilization and α-tocopherol release was significantly decreased in the elderly protocol, and that the gastric emptying should have been the key factor that modulated these kinetics. Having this information, we set out to produce tailored food products, using the explained before excipient nano-emulsions as a strategy to improve the spinach lipophilic vitamins bioaccessibility in older adults. To do so, spinach leaves were boiled or sautéed and then minced or blended along with excipient nano-emulsions, and their texture and rheology were analyzed. The samples were also submitted to an in vitro gastrointestinal static model adapted to the older adult population, and the excipient effect on vitamins assessed, as well as the cellular viability and antioxidant activity. So, analyzing the textural profile analysis of the minced samples, we did not see statistical significant differences. However, the addition of excipient in samples processed similarly lead to a small reduction of the parameters correlated with mastication. As for our sample processed into a pori, we do not have a comparison, but we can observe that parameters obtained propose this as an adequate food for dysphagic diets. Analyzing the vitamin concentration after older adult in vitro digestion, we can see that generally samples processed with the excipients and colors had higher concentrations of lipophilic vitamins in the whole digestion, with statistical significant differences. However, this cannot be said to the soluble portion, as we can see in the micellar phase. And if we look to the vitamin quantification overall, we can see an increase from 30 to 100% with the addition of excipients, depending on the processing type. However, and interestingly, in our soluble portion, we only saw an increase of 40%. And this can be due to the constitution of our excipient nano-emulsions, in which the low concentration of surfactant and oils could have impaired the solubilization. As such, because the solubilization was not increased in the same proportion, we did not see great improvements in the vitamin's bioaccessibility. Regarding the cellular viability, none of the processed samples with excipients present a negative effect on the cellular viability. However, we can see a tendency that the sautéed sample was at a lower cellular viability compared to the boiled samples. And theoretically, this could be done This could be due to the fact that sauté was not as effective in removing the anti-nutrients from spinach. Regarding the antioxidant activity, we did not see once more significant differences because the micellar phase was used for this analysis. Overall, from this study, we can conclude that the excipient nanoemulsion significantly increased the amount of available lipophilic vitamins in the whole digester, and that their composition directly impact the micellization process. On the positive part, we did not saw a significant effect on the cellular viability, but we saw that different processing techniques could affect significantly the digestive outcome. And overall, this type of strategy using excipient nanoemulsions is a very simple and viable strategy to improve the lipophilic vitamin intake in older adults. As a functional food, we proposed a novel functional thickened beverage enriched with vitamin D3 to produce a safer swallowing beverage with improved nutritional properties. And to produce this beverage, we extracted chia seed gum and produced solid lipidic nanoparticles incorporating vitamin D3, which were lyophilized for solubilization into different liquid matrices. These formulations were optimized to obtain the expected rheological properties, and then the selected formulation was applied into a dynamic in vitro gastrointestinal system composed by 4 reactors based on the TIM1, with the protocol based on the semi-dynamic and older adult model to mimic the conditions found on older adults to assess the vitamin D3 release and bioaccessibility. From the optimization of a formulation, we saw that the vitamin D3 concentration did not have any significant impact on the rheological properties of our beverages. And this is quite a good result because you can modulate the concentration of vitamin that we want to use. On the other hand, we saw that the matrix constitution, specifically the composition with lipids, carbohydrates, and fibers, significantly enhanced the thickening of our drinks. Finally, we selected a formulation with 0.75% of gum, and 0.25 of vitamin D3 to propose adequate rheological properties and the upper tolerable level of vitamin D3 for older adults. The rheological properties of our beverages were validated after their production, since all of them attain a nectar-like consistency, and they were planned for the in vitro digestion, in which we analyzed the vitamin D3 release kinetics. You can see that initially simpler matrices, namely the water in blue, had a higher vitamin D3 release concentration compared to the more complex matrices, such as the milk at gray. However, the more complex beverages increase significantly compared to the others, and this can be due to the gastric byproducts that could have influenced our nanostructure dissolution and the micellization of vitamin D3. Apart from statistical differences at the initial point, we did not see many differences throughout the digestion. However, if we look to the accumulated fractions of jejunum filtrate, we saw that the higher amount of vitamin D3 absorbed was in that reactor, and it was expected since our sample was complete. On the contrary, we can see that the beverage formulated with orange juice, which had a higher constitution of fibers, had a lower amount of vitamin D3 that was absorbed. Moreover, combining all fractions and calculating the total amount of vitamin D3, we saw that the simple matrix composed by water was not effective in preventing the degradation of vitamin D3 compared to the other two. Because all of this, the bioaccessibility was significantly affected. And I remind you that the only difference between these formulations were the selected matrices. And we can suggest that the lipids and proteins availability in the semi-skimmed milk could have improved the mixed micelles formation, hence improve the bioaccessibility. Regarding the cell viability studies, we did not see a negative effect in isolated cell lines. However, when we use the co-culture, we see a lot of significant differences. And once more, we can only attribute this effect to the matrix constitution that could have affected the transport and diffusion mechanisms. And overall, we could propose that functional tinted drinks are an interesting approach to provide safer products with improved nutritional properties for dysphagic diet and older adults. As conclusions of this work, we saw that overall, all nano-based delivery systems greatly improve the bioaccessibility of the study vitamins. However, we must stand out that excipient nanoemulsions were viable and very simple for food applications without the need to extract and purify our compound of interest. We also saw that gastrointestinal changes cause a significant impact on the digestibility of foods, and that the prolonged gastric emptying was the key factor behind all this. Additionally, we demonstrated that we should and can modulate functional and excipient foods to provide food with adequate properties and nutritional content for older adults. As perspective for future work, despite having used a lot of different in vitro models, it's necessary to establish better correlations between in vivo models to improve their fidelity. I also spoke a lot about micellization and solubilization. However, it's still quite unknown how these digestive mechanisms are occurring, so it's important to unravel them at reduced scale to understand how the nanostructures or other food components are interacted with our bioactive compounds in improving their bioaccessibility. Finally, it is important to develop more in situ models to propose non-invasive analysis of in vivo in specific populations or on humans overall. Thank you all for listening, and I hope that you enjoyed this presentation. Hello everyone, my name is Jean-Michel Fernandes, and today I will present the work.
Thank you. Thanks. Thank you for your presentation, Jean-Michel. We apologize again for the issues with the sound at the start. Just one question that I would like to ask you. You did really interesting and important work on trying to understand the differences between the digestion in adults and older adults. Could you point out the key differences in the in vitro protocols that you used to simulate the digestion of each of these populations and why this can impair the digestion and absorption of key nutrients?
Yes. So I use different types of in vitro systems. So when I did the static model, I used the recently published INFOGEST harmonized protocol. But then I also used that one to adapt to the conditions of the dynamic models that I used. So for instance, in the DITGI, we use the in vivo data that we have for the gastric emptying of yogurts. And then we modulated those parameters based on the recommendations proposed by INFOGEST. So on gastric emptying and reduced enzymatic activities. And what we saw there is, despite the fact that we have lowest amount of enzymes, the prolonged gastric emptying modulated the gastric phase digestion, so that consequently had a deep impact on the release of our bioactive compounds. So I think the gastric emptying, in this case, in dynamic models, was the key factor because it mimics better the conditions found physiologically in vivo. In the other system, I did not do the comparison study, but it was similar. Our system is based on the TIM1, but we do not have a computerized control of the valves. So we have to control the gastric flow and the passage of food and or chyme to different reactors. That's why we applied the semi-dynamic in vitro model to do that. And the enzymatic conditions were based also in the other adult model. So I think that most of all is the gastric phase that detects how the food will be digested and further down the line absorbed. The digestion is not as effective, but it's also longer. So we do not know for sure how that will play out, but it's interesting to study, I think.
So yes, so it takes longer to hydrolyze and break down the food structure and then hydrolyze the nutrients, and then
yes, and during that step, different byproducts can be formed, or soft aggregation can be occurring. I don't know. We did not look into proteins, for instance, when we use yogurt, but it would be interesting to see if the proteins were indeed easily degraded in the adult versus the other adult. So that could be a possible future work. Not only look at the bioactive compounds as we did, but also to the food matrix.
Macronutrients, yeah. Okay, thank you very much. I don't think we have any more questions yet. Thank you very much, Jean-Michel, for presenting your work. Congratulations again on defending your Viva just last week. And best of luck for the next challenges in your career.
Okay, thank you very much for the opportunity, and keep on doing this. It's very, very important.
Thank you. So just before we wrap up, I might just thank André Brodkorp for helping today and Didier Yeah, sorry about that. So next month, we are going to be live again with a presentation by Ana Blanco from the University of the Basque Country in Spain. And we are going to have two different speakers, Charlotte Oliver from Ireland, from here, Teagasc, Ireland in October, and Maria Santiago Vivanco in November. So if you are interested in presenting your work at one of our webinars, please send us an email at infogestviva@gmail.com. Thank you very much for listening. See you next month, and until then, enjoy the summer. Thank you.